Hello and welcome to our webinar today. I am Erica Ullman-Sapphire. I'm a professor in the Center for Infectious Disease at La Jolla Institute for Immunology and the incoming president and CEO. I have the distinct pleasure today of introducing our newest recruit, Dr. Sam Myers, and also introducing the Institute to you. So I'll begin with the Institute. There is a single word that drew me here when I came 2 years ago. And that is this, that is imagine. Imagine life without a disease. Imagine if you had spent this year without a mask. Imagine if you had spent this year going on the trip you meant to go on. Imagine for other diseases. Imagine if there was a way of finding out your cancer susceptibility early enough to launch your own immune system against it, to find and clear those single cells before they became metastatic tumors. Imagine if there was a way to find and reverse diabetes before it progressed. That is what drives all of our science here. At the La Jolla Institute for Immunology, we are a focused think tank. We're one of the top 5 in the world focused on immunology and the power of the human immune system to find and destroy invading pathogens and to harness the power of that immune system to prevent disease. And we study everything from disease. We like to say from Alzheimer's to Zika. We were founded in 1988, and right now we run 20 independent research laboratories, each one of them filled with postdoctoral fellows, graduate students, trained technicians, and career scientists. And each one of these laboratories is headed by a professor with a different research direction, different research focus. All of those are united in the study of the human immune system. And so by shining light on it from different directions, we can build collaborative networks and gain a better understanding of health and disease and how we can fight it. We run largely on federal funding that we compete for with NIH grants that powers our research, and that is our bread and butter. We have almost triple the national average. Our faculty are among the best in the world. But a lot of our funding also comes from private philanthropy. And that is key for launching immediate initiatives. So for example, if there's a mutation we're concerned about in the world, a gift can help us get that analyzed faster and get that data that we need to fight COVID-19 faster than we can if it takes us a year and a half to go through an NIH grant process. So both of those sources of funding are key to powering that discovery and leveraging what we do immediately and long term. We are divided across 3 different disease centers: autoimmunity and inflammation, how to control the power of the immune system and dial it back when it's become destructive. A center for infectious disease and vaccine research, how we can conquer the debilitating effects of viruses and other microbes and build vaccines that safely deliver lifelong immunity. And a center for cancer immunotherapy, how to use the power and precision of the immune system to seek out and destroy individual cancer cells. Among those 3 very broad areas, we target and analyze a number of different disease states with the goal of understanding what we need to do to harness and propel the human immune system. We are also the National Center for key databases, like the immune epitope database, anything you want to know about recognition by T cells and B cells and how to harness that data in order to develop better treatments and cures. We are also the headquarters for DICE, a database of immune cell epigenomes, how genes, individual genes and individual cells and individual people are modified to trigger disease susceptibility, or how we can use that information to develop better cures. We're also the headquarters for national and international collaborative efforts like the San Diego Center for Precision Immunotherapy, Together for Asthma, and 2 groups I direct, the Viral Immunotherapeutic Consortium and the Coronavirus Immunotherapeutic Consortium, where over 300 competing therapeutics are here in my lab being compared side by side to find out which are the most effective and why, and which are the ones that we need to launch for treatment. We translate our technology when we can. We have over 100 different licensing agreements, 5 drugs in clinical trials, and 3 cancer clinical trials ongoing. You have probably seen in the news the work that we did uniting a year ago across 8 different laboratories at La Jolla Institute to tackle coronavirus. And we've done that from everything from looking at antibody therapies to the effects of vaccines to teasing apart why some people get sick and other people are asymptomatic. What are the actual differences at the genetic level and the cellular level that control susceptibility to disease? And that individual nature of disease is a powerful area of discovery that we can use tools like Sam will tell you about today with our precision genomics or precision proteomics to understand the differences in different cells and the differences in different people that lead some to be susceptible to disease and others not. Our research is leading and has been picked up and described in a number of different news outlets and scientific press as what we are discovering here in these walls is setting the framework of what it is that we need to understand for this pandemic, future pandemics, cancer, diabetes, and heart research. So when I said there was a word that drew me here and drew Sam here, that word was imagine. With the most powerful tools commercially available that we have here, and the ability to dive into individual susceptibility at the cellular level, at the genome level, how individual genes are modified, how your own infection history and immune experiences control your response to diseases. That is what powers our research. And there's another word that drew us here, and that word is Mitch. Our president and CSO, Chief Scientific Officer, Mitch Kronenberg, that recruited me here and recruited Sam here. So after Sam gives his presentation, we'll bring Mitch back in and he will help lead a question and answer to address the things that you want to know. Right now, my distinct pleasure is to introduce Sam Myers to you. Sam is our newest assistant professor. He is a native Californian, now come home to build his research program. He got his bachelor's degree in biochemistry at Cal Polytechnic in San Luis Obispo, and his Ph.D. in chemistry and chemical biology at UCSF, where he studied O-glucanac signaling, so carbohydrate signaling in pluripotent stem cells. Most recently, he has been a postdoctoral researcher at the Broad Institute of Harvard and MIT, and a research scientist there too. And he's come home to us to build a research program here that bridges biology and technology development. Science is moving toward requiring more interdisciplinary approaches to research, how we can bring different tools to bear to untangle problems that no one has yet done alone. And Sam is a leader in understanding how to apply leading technology and that precision information to understanding a pathway that might lead to a disease or a pathway that might lead to prevention or a cure of that disease. So at the Broad Institute, Sam worked to improve the sensitivity of technologies like mass spectrometry. And that has allowed him to detect and study molecules that are in very minute quantities, because it can be the small quantities of things that can trigger to a disease state or not a disease state. When we have a better understanding of how, when, where, and why cells make the panoply of molecules and states, we can better study the complex series of events that lead to disease. And that's particularly important in the human immune system, because the human immune system is incredibly complex. It is designed to respond to pathogens we have yet to uncover. Here at LJI, what Sam is building is the laboratory of immunochemical circuits, understanding how that level of information and control of the information, control of the different molecules that are in cells can drive the development of autoimmune disease, cancer, and more. Now, another thing you may not know about Sam, he has a bit of an artistic streak, and he was an advisor to the artist in residence when he was at the Broad Institute, taught her how to make certain molecules that she needed, in fact, melanin, the thing that gives us our tans, and use that to make art. So Sam is artistic in person, artistic in his science, and we are so pleased to welcome his laboratory to LJI and learn all that he will uncover here. So Sam, I will turn it over to you, and we're looking forward to hearing about what you're building.
Thank you so much. That was a really sweet introduction. And I just want to say I very much appreciate the fact that I can be here and the fact that I am here and everybody came to this talk. What I'm hoping to do is share my vision with you and kind of what I'm striving to do, and how I believe it'll really impact the way we understand the immune system. And so I titled my talk, Observing the Immune System Through a Proteomic Lens. And you can tell how fresh I am here. I still have MIT in the background of my slides. I need to update that to my new location. But anyway, so what I'd like to talk to you today about is, again, using proteomics to study the immune system and all the insights that looking at it from a new focus or a new lens will lend us to study disease and how we can treat it. But I'll give you just a little bit of background about me. So I actually got interested in science at a community college in Fullerton up in Orange County. So that's where I grew up. And I took a human biology class with Brad Dawson, and with Brad Dawson and Chuck Lavelle really pushed me into science and really encouraged me to follow this. I finished my degree at Cal Poly San Luis Obispo, where I worked in Lisa Lindert's lab, and we studied kinases, or special enzymes I'll talk more about in a second, from Plasmodium, the malaria pathogen. And then from there, I really got my feet wet when I joined Jay Kiesling's lab at University of California, Berkeley. And we did metabolic engineering in his lab to make bugs make drugs. And this is where I actually got introduced to mass spectrometry and really fell in love with it through a scientist there named Chris Petzold. So when I applied to graduate schools, I wanted to go to a strong mass spectrometry lab. So I ended up going to UC San Francisco, where I was in Al Burlingame's lab, and he's the director of the mass spectrometry facility there, and he's one of the grandfathers of current day mass spectrometry and proteomics. But I also wanted to blend the technology that I was going to learn with the biology, so that I could speak both languages and understand what one hand is saying to the other. So I also joined Barbara Panning's lab, where she's a world-renowned stem cell biologist. So my background, I applied a lot of these technologies to study stem cells and how stem cells differentiate and make decisions. And from there, I wanted to get more of an immunology background. So I went to the Broad Institute, where I did my postdoc, and I was an actual research scientist there for a couple of years. So again, I joined Stephen Carr's proteomics platform at the Broad, where again, we push these technologies, and I worked on technologies to make the readouts we have more sensitive, so that we can actually study real systems, and rather than cell cultures or things that don't necessarily always reflect true fidelity of biology. But then I also was in Aviv Regev's lab, and she was a fantastic advisor who showed me a lot about immunology and computation. So I learned a lot from this kind of growing up in the scientific community. But now I'm back in California, so let me tell you what I'm actually thinking about these days. So this is kind of a classic video. This is a classic video David Rogers took back when he was at Vanderbilt in the 50s. And what we can see here is we can see this neutrophil, and it's chasing this little black dot here, this bacterium. And you can see that it's basically ignoring all the other red blood cells that it sees around it. Right? So one of the things that's important to know about this is that our immune cells are always surveilling their environment. They're always out to see kind of what's happening. And how does the cell know to attack this particular bacterium while ignoring the blood cells of its own counterparts around it? And so one of the things I think about a lot is how we're really good at understanding the decisions cells make when they see something like that. So for example, you can have some kind of precursor cell. So let's say it's our neutrophil that I showed you in the last slide. It can have some input or some signal like a bacterium, and it'll turn on a certain response gene or set of genes that will change it to this different cell state. And we usually look at these as what we call heat maps, and we just stare at these like magic eye paintings and hoping to see a pattern. But what we do is we can say, in one cell state, when the cell's not activated, some genes are off and some genes turn on. And in the activated cell state, when there is a bacterium present, we can say, oh, there are genes that are turned on that weren't turned on before, and then they also shut off a bunch of genes. And so we understand this decision that was made by the cell. But one of the things that I'm particularly interested in is the information processing that goes on to make that decision. Or how is the cell actually thinking and understanding and interpreting its environment when it sees something like a bacterium or a red blood cell that it wants to ignore, or the latter. And it goes through this information, these circuits, or these biochemical circuits, or what I call immunochemical circuits in the phrase in the immune system, where somehow it's processing this information, and it says, okay, we know that we need to turn on some specific genes to attack this bacterium. Or it can go through different routes and say, oh, the way we're interpreting this is something that we don't need to attack, right? And it would be really great if we had a really well mapped out understanding of how information is processed in cells. But I would actually argue we're severely lacking in this case. We know kind of some of the major landmarks, and we know some of the pathways that these cells use to make these decisions, but we have a lot of missing information. And this is a lot of space that we can actually use to develop therapeutics for if we could learn how to intervene. And so I'll take you back kind of, so to put you in my frame of mind, or the way I think about these things, I'll take you back to the central dogma of biology, right? So we have our DNA, which is the molecule that makes up what you've heard called of our genome. And certain genes of our genome are transcribed into mRNA or messenger RNAs. And sometimes we call this transcripts. And when we look at all the transcripts at the same time, we call that transcriptomics. And then these mRNAs are translated into proteins. And then when we can look at all the proteins, so what I do is I look at all the proteins in the cell, and we call that proteomics. So usually omics is a term meaning we're trying to look at all things at once so that we don't miss anything or there's nothing really in our blind spot. And the reason this is important because depending on what type of molecule you look at, you get a different sense of what's going on. So the way that I like to think about it is the DNA is basically all the possibilities where all the choices a cell can make are stored in this, information storage molecule of DNA. Some selections are made and the information is then transferred from the DNA so that it can eventually be translated into proteins. And actually, that is where the functional aspect comes in, or the execution of these functions. And proteins do all sorts of things in the cells. They not only create scaffolding and structure within the cell, but they do small chemical reactions like metabolism or signaling and internalize signals and sense things. So really the functional parts of a cell is the proteins. So we really want to understand what proteins are present. And I'll give you kind of an idea of why this is. So I think in 2019, I went to 9 or 10 weddings. So this was on my mind a lot is, someone would ask, how was the wedding? Well, if someone asks you how the wedding was, you could tell them all the possibilities of everything that could have been decided, right? You could say, oh, we had a large number of dates that we picked through. We had a handful of venues. We picked the drinks and the food, and we had our songs picked out. And then we had our guest list, right? And the bold line people are people we really wanted to invite. If there's room and space, we invite the smaller line people. And then there are a couple people that aren't going to make the cut, okay? But this is all the possibilities that the wedding could have been like when someone asks you, how was the wedding? Where you could learn about the selections, right? Where they say, oh, we picked a certain date, and we have a venue and then a backup in case it rains. And we pick these 3 types of beverages and this 2 meals. And we have our song list. And these are the people that we've ended up inviting and have RSVP'd. But if you really want to know how the wedding was, like if it was fun or what was going on, you want to know what the actual turnout was, right? You want to ask about what the turnout was. So you know the date, it didn't rain, so you had a good venue, but the caterer showed up with too much of this pink drink and brought a different type of meal, and the food was great, but your guest list was all over the place. Your red friend actually didn't show up, and the dark blue friend that you actually didn't end up inviting showed up with extra people, and someone brought a kid and a new partner, and you had people that you didn't even invite crash the wedding, and they brought their friends, and they brought a dog. I mean, who crashes a wedding with the dog? Anyway, the point of this is saying that where you look at these different biomolecules is what kind of you understand about the system. So again, I would relate this back to the genome. Again, is all the possibilities. The transcriptome is kind of what's been selected, and the proteome is what's actually happening right now and what are the activities that are occurring. And just to take it back a little bit, kind of more simple analogy is if you think about a caterpillar and its metamorphosis, a caterpillar and its metamorphosis into a butterfly, it's good to know that these actually have the same genome. But if you want to get the full picture of what the insect is actually doing, you want to look at the proteome, because then you'll see the difference between these two. The genomes will be the same, but the proteins present will be vastly different between the two. And it's really, so I show you this DNA to RNA to protein in this linear sequence of events. And it's actually a lot more complicated than that. When scientists first sequenced the genome, we thought with the complexity of our species, of the human species, that we had to have up to 140,000 genes in our genome. But it turns out we only have about 25,000. And so where all the complexity comes from and all the diversity comes from is the proteins and the activity the proteins have. So DNA folds in on itself and compacts it. You know, you have 2 meters of DNA in every cell, and it's all compacted into cells and not randomly. In fact, we have labs at LJI that study this and how this influences gene expression. But this DNA can package itself, and it's actually proteins that, you know, pack it and hold it tight. As DNA is transcribed into RNA, there are proteins that accelerate this process, so there are proteins that actually slow down this process. The mRNA itself can be stabilized by certain proteins, or it can be rapidly degraded in case it's not supposed to be there anymore and you don't want it translated into a protein. And then protein translation is also mediated through proteins, and its activity can be increased or decreased depending on which proteins are there. And the proteins themselves control other proteins. So a protein, you know, can be destabilized and degraded, or it can be it can have little modifications added to it that make it last longer. These chemical reactions that these specific proteins do, they do specific chemical reactions in our cells that can be sped up or slowed down depending on how many molecules of this protein there are, or how it's modified, or how its activity is adjusted. So the protein in the cell is really doing a lot of the work in here. So it's really important that we actually look at proteins and the modifications to proteins. So what do I mean when I say modifications to proteins? So something I study a lot is they're called post-translational modifications. And I think of them as like chemical ornaments on proteins. So you might have your protein of interest here, and it can be glycosylated, and a small sugar might be added to this protein of interest, or it can go down a different pathway, and then a phosphate can be added to it. And a phosphate is just a small inorganic molecule that can also be added to your protein of interest. Or, you know, potentially your protein goes through both pathways and is modified by both the sugar and the phosphate, right? So this gives us a lot of diversity in the proteome, and why this is important and why it's important that we study this is that each one of these species is a distinct molecular species, and it might have a distinct molecular function. So again, it might catalyze a metabolic reaction faster or slower. It might bind DNA to turn on specific genes or turn off specific genes, right? So, you know, how these post-translational modifications change, or how these chemical ornaments change proteins, it's really important for us understanding what the cell is doing and how it's actually, you know, using these to process information within itself. And I'll go back to the wedding analogy for a moment, where these chemical ornaments regulate activity and what other proteins they interact with. So again, let's say you have a handful of guests at your wedding, and you know, people are having some cocktails, and some people brought their kids, and a couple folks brought cigars, and so they can, you know, as a celebratory thing. These people won't randomly assort themselves throughout the wedding, right? Throughout the party. What they'll actually do is they'll cluster together based on how they're modified or what chemical ornaments they have, right? So the folks with kids are probably going to cluster with each other and, you know, let the kids play with each other, when they're probably going to cluster away from people smoking cigars, right? And then what we can do, so what I do is I develop ways so that we can identify these modifications and identify which proteins they're labeled for. And by understanding who they're interacting with, we can understand the function of it. So you might say that the people with kids go to bed early at the wedding, and the people that with the cigars and all the martinis are staying up too late. So we can infer activity by which proteins are in complexes with other proteins, and how these modifications drive different interactions and different activities. So let's go back to the neutrophil that I showed you at the beginning, where we have this high resolution zoom in here. So how does the neutrophil know to chase this bacterium in this direction, but not in any of these other directions? You can see it's kind of protruding out here, but it's actually knows, in the video, it knew to move this way. And as this bacterium swims away, it leaves behind it kind of like a scent. It leaves behind these small molecules, or potentially inflammatory molecules, that leave behind it in kind of a concentration gradient. And the reason our immune cells can actually chase it in the proper direction is it has proteins embedded in its membrane that can bind and sense the presence of these molecules. And once these receptors sense a ligand or sense a molecule like this, it will transduce a signal through post-translational modifications into the inside of the cell. And so what I'm showing here is basically you're getting a large signal from a large concentration of this inflammatory molecule. Over here, your receptor is picking up a little bit of it, so you get a little bit of the signal transduction occurring here. but this molecule is not detecting any of it. So the cell knows by changing these post-translational modifications in this direction, it knows to actually chase in this direction towards the molecule itself. And this is a very central process to the immune system. You know, it's not only bacterium, this bacterium that I'm showing, it's also fungus and worms. They leave a scent behind them that our immune cells can sense, but also our cells that are virally infected. So our cells, when it's infected with the virus, will display small portions of proteins, what we call peptides, on the surface of themselves. And our immune system will come and check out those peptides displayed on the surface and say, what's going on inside of you? Is everything okay? And if it sees something that it doesn't recognize, like a viral protein or a viral peptide, it knows to attack it. Same with cancer cells. So cancer cells are, you know, just cells of our body who have decided to no longer cooperate with the rest of us and the rest of the cells in our body and use them in nutrients. And they will also have certain things about them that something's off. There's a mutation in the genome that translates itself to the protein, and that protein can be put on the surface. And a lot of cells, like T cells, for example, can recognize that and destroy it, and destroy that cancer cell. But immune cells are often getting mixed signals, right? It's not always, you know, attack or follow or destroy, right? There are signals that negatively regulate these processes, and these are important to understand these as well, because, you know, on our bodies and in our bodies and on our skin, you know, there are 10 times more bacteria and fungi on our bodies than our own cells, and we tolerate them, and they're actually commensal for us. They protect us from bad pathogens, certain bacteria. They supply some of our nutrients like vitamin K. So it's important that we get some signals to say, okay, don't actually destroy this bacteria, so we need to find this balance. And the same is with cancer. You know, like I mentioned, cancer is just some cells of our own that have decided not to cooperate anymore. You know, so we want to be able to recognize the mutations of a cancer cell and say, you need to be destroyed, but we have to balance that with looking at our own healthy tissues. We don't want to attack our own healthy tissues, and this is something that occurs with a lot of autoinflammatory diseases. So there's a balance that occurs, and it's these mixed signals that immune cells get that's really important for us to understand. So this is where proteomics really shines, is in this kind of the signaling and the signaling transduction. So the first thing that we do as a proteomics community is we can inventory these cell types. We can analyze these cells and say, do we detect these certain receptors? And if we do detect these receptors, we can say these are probably important, or it's probably important for this cell because it probably detects this inflammatory molecule, right? And that's good for us to know. That means we can assign a function to this cell. We can also see if it's recognizing something specific, like a bacterial scent or a worm scent. But what's really powerful is we can watch these changes over time. So if we introduce a bacterium to this immune cell and we see it turn on more and more of these green genes or these green receptors, then we know we can infer that these are probably important for detecting this bacteria. And then if these bacteria are there for a lot longer, they might start turning on more of these negative regulating receptors. And we can infer this as, you know, it wants to shut itself down. It doesn't want to remain active forever because that would be dangerous to our body too. But what I'm particularly interested in is all these signaling pathways and the post-translational modifications that change when an immune cell senses either a healthy tissue or a diseased tissue or a pathogen. You know, in the first, you know, within 30 minutes of introducing a bacterium, there are thousands of post-translational modifications that change to proteins within a matter, like I said, of minutes, right? They can be added or removed from these proteins to change their activity. And one of the things my lab will study is how does the cell interpret when it's getting mixed signals? So again, if you're getting a little bit of signal A and a little bit of signal B, how do you know whether to attack or whether to stop attacking? And this is something that mass spectrometry is well suited to study. And then again, we can assign molecular functions to a lot of these proteins by looking at what they interact with. So if we can tell that this green receptor is interacting with this inflammatory molecule, we can infer that it's probably sensing that particular molecule. Or like I mentioned before, if the cell is presenting peptides on the surface and says this is a snapshot of what's happening inside of me, you know, make sure I look healthy. You know, we can discover how different cells communicate with one another, whether it's direct contact or sending out their own signals. And so the title of this talk, or the title of this seminar overall, is, you know, the emerging field of proteomics. And I think I would narrow that down a little bit more. This is especially emerging in immunology. And so I gave you all these applications for proteomics. Why is it not used more in immunology? Or, you know, why am I just starting now? Well, it's a lot more straightforward to get information out of DNA and RNA because they're information molecules. So it's easier to copy and store that information. With proteins, they don't lend out the information very well because, again, they're functional. So basically what we do is we just weigh them to see how many proteins we have of certain types. And so getting into a little bit of the chemistry here, so we can, if we know the molecular weight, or if we know the elemental composition of a particular analyte or component, we can estimate by its weight how many molecules are there. So here I'm showing a gram of sugar, of sucrose, balanced on the scale, and we know the elemental composition of sucrose. We know what these carbohydrates and these sugars look like. And from that, we can calculate that in 1 gram of sucrose, there are 1.8 times 10 to the 21 molecules of sugar in there, of sucrose in there. And 10 to the 21 is basically laid out down here at the bottom. So you can see we can measure, this is a huge number of molecules, right? But we can't measure things at this gram scale all the time. So what we use is we use a mass spectrometer. And mass spectrometers are very good at measuring minuscule amounts of mass and of matter. And through some generous philanthropy, the Institute is going to be able to purchase this Orbitrap Fusion tribrid mass spectrometer. And what I mean by tribrid mass spectrometer is this is kind of the Swiss Army knife of mass spectrometers. It can do all sorts of things. And I'm not going to take you through everything, but this is just kind of showing the bare bones of where a lot of the chemistry and physics. And I think these are fascinating to me because it's such an engineering and chemistry and physics feat to get these to work together. And these aren't huge instruments. So for scale, I'm showing a former colleague of mine, Dr. Klager, and you can see that it's about, you know, the size of a person. You can fit it on a desktop, so we can install these relatively easily. But where the action really comes in is this thing called the Orbitrap mass spectrometer, or the mass analyzer. And this is actually quite tiny. This is the size of a nickel, or you can see the comparison to a nickel here. And the sensitivity of this is really great. So we can measure femtograms of material. So a femtogram is if you divide a gram by 10, 15 times. So if you remember that long stretch of zeros that I showed you before, so we can actually measure about 2 million molecules rather than 10 to the 21 molecules. So very minuscule amounts of protein. So how does this give us any information about? Oh, actually, let me show you a little video of how a mass spectrometer works. So first, our analytes are electrosprayed into this vacuum in this capillary, and that was actually a huge feat in of itself. John Fenn got the Nobel Prize for that in 2002. And as our analytes are introduced into this vacuum, we apply different electrical currents to these rods that you can see in the corners. And basically what this allows us to do is push these positively charged analytes into the mass spectrometer, where we collect them in a trap and we cool them down by slightly bumping them into nitrogen gas. And this allows them to kind of behave more as a unit. And then what we do is we inject them electronically into the Orbitrap itself, where they orbit the center electrode here in a speed that's proportional to their mass. So think about a bowling ball and a tennis ball being on strings. If you were to swing those, they'd swing at different speeds, right? And then using some fancy math called Fourier transform math, we can actually process this signal into distinct masses. So this is how we measure things in Orbitrap mass spectrometer. So how do I get any biological information out of this? Using, you know, measuring proteins or measuring these modifications. So if we have, again, a protein of interest and we know it has a certain mass, if we measure it and it's a mass of plus 80, we know that we can infer that there's a phosphate on there, because the molecular weight of a phosphate is 80 Daltons. If we measure the mass of our protein of interest and it's an addition of 203, we know that it's probably a sugar that's added on there. And the reason my lab is going to study the interaction and the mapping of these post-translational modifications in this manner is that kinases, for example, are great classes for drug targets. I mean, most of the drugs out there that are clinically approved either directly inhibit a kinase, which is the protein that transfers these phosphates, or somehow inhibit a pathway that a kinase is involved. But what I'm going to add another level of that too is the potential for this glycosyl transferase that transfers this sugar. It's actually a really good potential for a drug target, but we need to understand it better. We know it's involved in a lot of things. We know it's important for a lot of disease progression, but we don't really have a good understanding of how it works biologically. And that's where I hope to come in, especially in the context of the inflammation balance and by T cells getting, or immune cells getting mixed signals. So one of the examples of the projects that will be going on in my group is that we'll look at the certain type of T cell, and when it's activated in the presence of this protein called IL-6, it becomes a T helper cell that's important for getting rid of bacteria and fungal infections. If the same cell is activated in the presence of this different protein called TGF-beta, it will become an anti-inflammatory T cell that will tamp down our own immune response. But if the same cell is activated in the presence of both of them, they become a very functionally distinct cell type called a Th7 T cell that is actually a pro-inflammatory cell. So these create inflammation and orchestrate our immune system to be inflamed and go crazy. And these Th17 cells have been implicated in a lot of autoimmune disorders like lupus and rheumatoid arthritis, and inflammatory bowel diseases, even multiple sclerosis. And there's even some evidence that it might be involved in type 1 diabetes development. Right? And so what my lab will do is we'll study the signaling pathways of how these cells decide whether to turn into an anti-inflammatory T cell or pro-inflammatory T cell. And we understand the kind of the landmarks of the pathways themselves. What we're really missing is how these pathways interact with each other. And people have gone looking for them, and it's remained elusive, but I'm uniquely qualified to apply proteomics to this, so we don't have any bias, and we can actually start to look for these molecules that are mediating the interactions, so we get a pro-inflammatory state. And the idea would be is that if we could identify these proteins that are important for turning on the inflammatory state, we could potentially drug them and push ourselves from an inflamed or pro-inflammatory state to an anti-inflammatory state. And this is just the work I want to do in my lab. One of the reasons I'm so excited to be here is all the collaborations I want to be a part of at LJI. So, just to name a few, are working with Susan Shreston and Erica Sapphire, looking at how viruses hijack our own cells and hijack the proteins in our cells to replicate and make us sick or cause disease. And we're going to specifically look at Ebola and Zika, and of course we need to understand SARS-CoV-2 better, the coronavirus. With Mick's Croft lab, he has a special interest in inflammation of the skin. He has projects where a cell can see one or the other signal and they don't respond, but if they see both of them, if they get this mixed signal, they create this inflammatory protein that is associated with these dermatitis and psoriasis phenotypes, disease phenotypes. Erica mentioned that LJI has the largest database of the immune epitopes, which are, again, are these peptides that get presented on the surface of cells so that cells can interact with the immune system and say, am I healthy? And this is with Alex Sette and Bjorn Peters. With Anjana Rao, she actually has a portion of her lab that studies this glycosyl transferase, but more in the context of how it influences DNA and how DNA gets damaged and mutated in the progression of cancers. So her and I are going to be able to collaborate in a lot of things there. And Patrick Hogan, and he studies how specific proteins called NFAT and AP1, how they bind DNA. And when they bind DNA together, they turn on the genes that a T cell needs to destroy a cancer cell. But if NFAT doesn't have AP1 as a partner, or it has a partner that we haven't discovered yet, the T cells, they don't recognize the cancer cell and they don't attack the cancer cell, and they become what's called exhausted. So, one of the things I want to help the Hogan group do is identify the proteins that are binding DNA as a failure to respond. And so to wrap up, I wanted to show you that, I've come from these two big labs with a ton of people and, a wealth of knowledge and capabilities and capacities where, here I am at UCSF, and I think this was 2008, and this is a couple years ago at the Broad Institute, where, while it was exciting to be part of a big group, I get to start something new. So here's a photo of me of all my lab, and all the people in my lab and all the equipment that I actually own, which is just those couple of boxes right now. But I'm working on, we're going to get some funding and we're going to expand, and hopefully I can build a team that was as exciting to be a part of as I had growing up in the scientific community. So with that, again, I just want to say thanks, and I'm happy to take questions or clarifications if I can. So I think next we're going to hear from the current president, Mitch Kronenberg.
Yes, thank you very much, Sam. That was great. We're really happy that you've joined us, and you obviously have enormous technical capability and knowledge, but also you really have a knack for explaining things that I've never seen that side of it before, and those analogies are very important for understanding, for example, how proteins come together to act as complexes. And those complexes of proteins can drive signaling, or they can drive cancer. And yeah, my camera is on. I was asked to put my camera on. There you go. So you've done a really beautiful job. I don't think I'll ever forget about the babies and the cigar smokers. So I just want to emphasize one point that Sam made, and that is that Sam's technical expertise in identifying the decorations on proteins and their interactions will help many labs here to advance their research. He has his own research program. He told you about the inflammatory versus the regulatory program of T cells. That's very important to many at the Institute. But he'll also be helping others. One example, or to provide another example, is we have an effort to identify tumor antigens, the mutations in cancers that need to be that can be recognized by your immune system, that can provide a handle for your immune system to fight a cancer. All cancer cells have mutations, and mutations are only changes in proteins. And some of those mutations provide a handle, if you will, for the immune system to grab onto. We have a program to identify these so-called cancer antigens, but Sam's proteomic techniques will accelerate that effort, not only in cancer, but in other diseases as well, in autoimmunity, and of course in infectious diseases. And I won't go over the whole list of projects that Sam will be involved in. So he's a critical player here. I want to emphasize that as the president, I could not have recruited Sam without philanthropic gifts. The mass spectrometer and its associated instruments that go with it is well over $1 million in cost. And we obtain those funds through philanthropy and startup funds for a new investigator like Sam to begin his lab, to get data that will provide the foundation for getting grants. Those funds are being provided by philanthropy. And we have more need in that area. So I think that's very important for you, our supporters who are listening to this, to understand that message. Okay, so we're going to have a question and answer session now. And I'm going to start, Sam, by asking you, I can have my genome sequenced for maybe $500 or $100. Can I have, will it be possible to have your proteome done at some point?
Oh, absolutely. I mean, there's kind of two ways to think of it. One is we can actually use your specific genome with proteome to get even more kind of precision analysis of what's going on there, right? We can look at your specific mutations and we can see how they're incorporated into your proteome. But the proteomics is good for a lot of other things too. So your blood, if we could take longitudinal samples of your blood, we can kind of understand what it looks like when it's healthy and what the proteins are when you're feeling ill or feeling great, we can start to see kind of what the balance of proteins are there. So yeah, there's all sorts of different proteomes that would be helpful and useful, I think, clinically to measure. So, and
Is
we
there,
only need a drop of blood, like a prick, finger prick. So
yeah. The amount of material is not limiting. Is there a database? If I have a protein, is there or a protein description that lists all the molecules that it will interact with based on earlier studies?
Oh, that's a great one. Yeah. So there's a couple different databases. So some labs have derived databases where they pull on a particular protein of interest and they see which ones come down with it. It's like a guilt by association, so that we can kind of understand the function of these. There are a couple databases like that. There are databases that store all the post-translational modifications that happen to individual proteins, and some of them have hundreds to thousands per. What we don't have a sense of yet, we know it happens in select cases, but we don't understand how those two influence each other. Right? We can have the whole list of PTMs and we can have the protein interactions, but we don't know how the PTMs specifically change specific protein-protein interactions. So that's something that hopefully we can bridge the gap as we kind of think about applying these more and more. That's kind of the point of why I'm calling my laboratory Immunochemical Circuits, right? So it's the interaction between immune proteins and how they create these circuits to process information. So that was kind of the driver behind that. So
one more related question. What about modeling? Do you think in the next 20 years, I mean, you have a long road ahead of you. Do you think modeling will be taking a bigger role in predicting how proteins interact?
I would say in the next 20 minutes, it would be helpful. Yeah, so in fact, I have some collaborators at the University of Pittsburgh who, what we do is we measure these changes in phosphorylations across the entire proteome, and we measure them in time. And then what we can do is we can build models to say, what are the enzymatic reactions that are happening the fastest? And then we can correlate that with what genes are turned on specifically. With the specific idea is that if we have 10 genes get turned on, but we have one that's problematic, if we can build a model to turn off that specific pathway, to turn off that specific gene, right? We could then have very precision therapeutics to control cells and how they behave and how they're thinking. So yeah, no, I think, yeah, I wish, yeah. Actually, one of the first people I want to hire is someone with computational background like that. So
great.
Got anybody in mind? Let me know.
I'm not going to share with you. No. So the next question or questions could be for both of you, Sam, and for Erica. And it basically is, does the work you do distinguish between vaccine-induced immunity versus natural immunity from an infection, or basically how can the work be applied to the analysis of the response to infections?
You want me to go first?
Sure, go ahead.
I think one of the ways I can immediately think of it is, again, these peptides that get displayed on the surface. If we were to look at our own immune system and we could identify these peptides, we could say, is it from something that we vaccinated with? So the spike protein from coronavirus. Maybe if someone has displayed peptides from the spike protein, we can say, oh, that's what the immune system is using. If it's using a different protein in the virus that we didn't use to vaccinate, right? Then that would tell us that it's more of that would allow us to kind of infer better that it was maybe an immune response to the endogenous virus rather than a portion of it, or an attenuated version like we do in the vaccine. So that's how I imagine I would approach it, at least at first.
And then we would characterize, what is the immune system targeting? If you had a previous common cold coronavirus, did that train your immune system to go and focus on a certain part of that spike protein? Is it shared with the new virus? Is it shared with an emerging variant? And how does that history of prior exposure steer how you're responding to something now?
Great. So Sam, what are the greatest barriers to faster progress in proteomics? I mean, you said you need 2 million molecules. When are we going to need 100 molecules?
Oh, that's
I mean, it's a big step from 10 to the 10 followed by 21 zeros to followed by 6 zeros.
Yeah.
But what are the barriers to progress?
So I think one of the things, there's some just pragmatic things. So like the mass analyzer that I showed you, they can make those more sensitive, but they just can't fabricate them consistently.
Mm.
Right? So just some things like that. There are limitations in the way we process our samples. So with mass spectrometry samples, they have to be very chemically clean. We've come a long way just in the last 5 years, how to get around having detergents and things like that, and it's too salty. A lot of my work has been done with minimizing sample processing, because every time you do a process, you lose a little bit. And so basically kind of one of my postdoc projects was streamlining all of that so that we handle it as little as possible. The speed of the instruments always are improving, and I think that always helps us, so we can do analysis for shorter amounts of time. So there are some pragmatic things, and I think just the inference tools and the analytical tools that we have too, the more like on the computational side. I mean, genomics has been so strong for so long that there's so much, there's such a wealth of information and an understanding of how all these things work together. We don't have those same databases and tools and computational tools that we do for proteomics. So I think a lot of those need to be ported over so that we can just interpret our data better. Maybe we don't need to measure 10,000 different things. Maybe we only need to measure 1,000 things really well, and we can infer what's happening there. So I think there's a lot of places that we can improve.
I have some questions here are a bit technical, like about sample collection and input into the mass spectrometer versus other analytical techniques like GCMS. I don't know if that's maybe that's a question to be asked offline, but
I'll just say that the difference, the main difference is GCMS is a lot smaller molecules. Proteins are quite a bit bigger.
Yeah.
We do a different kind of, the C is the chromatography, and it kind of on the fly purifies things, and it's just a different way to purify things. We use liquid chromatography, whereas small molecules like sugars are typically done with gas chromatography.
Yeah. Do you think, and Erica, you can chime in too, do you think proteomics will help with personalized therapies, personalized immunotherapies, for example?
I can't see how it could hurt, right?
Right.
Sam's technology is to dive into the complexity of things at the small level. We know that with immunology and human disease in particular, one size does not fit all and never did. Your immune response will be different from my immune response. Some people get very sick, other people are asymptomatic. You have a unique prior history. That prior history, and in my lab, how viruses control and conduct your cells affect all of those decorations and modifications they put on those proteins. Sam has the tools to untangle that and figure that out in a way that we didn't have before.
Sam, do you want to add anything on the personalized immunotherapy front, or?
I mean, yeah, I think there's a lot that we need to just understand about, how our different cells react in different ways, and our mutations in our genome that are normal and that create our diversity. They can lead to different signaling, and why are some people getting very sick with COVID, and why are some people not getting good? Right? That's probably has something to do with the signaling and the cytokines that our cells produce. So, it'd be really great if we could start understanding, the normal mutations the normal variants that we have in our genome, and how that translates to like the actual functioning of a T cell or of an immune cell. So yeah, I think there's a lot there to be done. So.
Yeah, I have another question. And how would you have added to La Jolla Institute's important work on COVID-19 and the pandemic if your program was up and running at the Institute last year?
Oh. Well, I mean, I think one of the things we could have done immediately is just how are the viral proteins hijacking our host proteins? Which ones are they hijacking? How are they hijacking them? How is that I think would be one of the first things. In fact, Erica reached out to me if we could do anything right away, and this was kind of the first thought I had, is like we should figure out how the viral proteins are interacting with the human proteins and hijacking them for their own good. I mean, that's something that I think is very crucial to be done well and to be done early. So that was something I would have loved to sink my teeth into then.
Great. Well, Sam, I really want to thank you. I know you're busy setting up your lab, for taking the time to talk to our audience who appreciate so much the work that's being done here. Thank you, Erica, for the great introduction. I just have a few more comments, and I believe there are slides that go with them. So the slides will appear at some point. All right, I don't see them, but that's okay. So here we go. Okay, so I just want to tell people two things. And first, we have another webinar coming up, and it features Sonia Sharma, and she'll be talking about developing more effective cancer immunotherapies. And what Sonia has done recently is looked at all the metabolites, the small molecules in the blood, and gained great insight into why some people get what are called adverse events to cancer immune therapies, to therapies that stimulate your immune system to fight the cancer. Some people get IBD or diabetes as a side effect. And Sonia's work, among other things, is helping us to understand what molecules are causing those adverse events. And if we know that, we can help to prevent those. She's a terrific speaker, and I highly recommend that event. And next slide, please. So I want to thank you for your attention. I brought up something about this. If Sam had been with us last year, Sam would have been with us last year if we could have afforded the mass spectrometer. So that's a very poignant illustration of how important philanthropy is for progress. We're going to keep pushing so we can move toward life without disease. That is our mission. And it depends, of course, on federal funds from the National Institute of Health through a competitive process. Our bread and butter, as Erica told you at the beginning. But philanthropy is very important. And philanthropic dollars go into certain categories, like helping set up a new lab or helping buy new instruments that we can't replace with NIH grants. So if you have the interest and you have the means, please think of us. And to support our research, you can go online or you can contact Chris Lee, our Chief Advancement Officer, and you have the contact information right there in front of you. Thank you for your attention. You've been great. Your questions have been great. I appreciate your participation. Thank you again, Sam. What can I say? It was terrific. Bye-bye, everyone. Take care.